All right, so we're going to look at some pea stuff and maybe some beetle stuff and basically get into Punnett squares and um, the probability of inheritance. So when we're talking about Mendel, a long time ago, grew a whole bunch of plants. And what he ended up doing is he found varieties that were true breeding, which means that they produced the exact same type every single generation. So you would call those like purebreds. And that was in this pea parent generation. And what Mendel would do is he'd take two different varieties that were both purebreds, have them produce offspring, and this offspring would be considered the F1 generation. What he found is these, when they're as long as they were mated with themselves, always produced white. These would always produce purple. When you mix them together, you end up with this first generation, and they all show one type. But then if you took those and went and self-pollinated these with themselves, you would find that there would be a 3-to-1 ratio of purple to white in the next generation. So mathematically, he worked out a couple different things here. We'll look at this from a Punnett square standpoint. So again, purebred round, purebred wrinkled, when they were crossed, all of the phenotypes in this next generation were all round. But then when all of these round were mixed together, mathematically found this ratio was about 3-to-1. And this is the basis of coming up with Punnett square work. So let's see what that looks like. Okay, so if we're going to say this is the genotype for purebred round, and this is the genotype for purebred wrinkled, what you would do is you'd look at, this would be the diploid parent, and you'd try to consider all of the different possible gametes that could be produced. So it could pass on this chromosome or this chromosome. This one could pass on this or this. And these are possible gametes, and these would be all of the different possible zygotes that could result from fertilization. So you'd take this one and this one, this and this and this, and those two. Now, even this would now represent heterozygous genotypes, but the phenotypes of all of the F1 generation would be round. So then what you would do is you take this F1 and cross it with each other and you end up with F2. So let's start this over again. So now when we're looking at, that's F1 and that's F1. One gamete could be that, the other gamete could be that, one could be this, one could be that. And now as we cross this up into possible zygotes, the ratio of in the F2 generation of this monohybrid cross would be 3 round to every 1 wrinkled. And even though in that F1 generation right here, you don't see the wrinkled varieties, in the next generation you do see wrinkled. So when we come back here, that's what Mendel saw. But then instead of just looking at one trait at a time, monohybrid, he'd start looking at dihybrid crosses. And again, he'd take these pure breeding varieties And he'd take these round yellow, which we'd represent with this, and wrinkled green. And again, this would be our P with the parent purebred generation. When you mix these up, you end up with all heterozygous offspring. From that, you could breed down to an F2 generation. So again, let's take a look at what that looks like in a Punnett square. Okay, so we're going to start with our purebred round yellow. And this is going to be our P cross. So we could do wrinkled and green. Now, when we split these up, a lot of times when people switch from monohybrid crosses to dihybrid crosses, they see that there's 4 boxes and there's 4 letters, so they just put an R here, an R here, a Y here, a Y here. That wouldn't be right. You need to have 1 pair of these chromosomes and 1 pair of these, because this is diploid. It has a backup of the R and a backup of the Y. So when it becomes haploid, you still have 1 of each set of genes. So you could have this R and that Y. You could have this R and that Y. This R and that Y. Or this R and that Y. 1, 3. 1, 4. 2, 3. 2, 4. So again, even though the we have the same genotypes here, they still are different combinations of chromosomes. So then when you put these together, every single one ends up like so. Again, we'd have 1 big R, 1 little r, 1 big Y, 1 little y, and every single one of these 16 would be heterozygous. So the F1 generation would all be heterozygous round and heterozygous yellow. Now, as we start getting into our F2 generation, things would end up looking a little differently, because you would take heterozygous and you'd mix it with heterozygous. So now we could go 1, 3 as a possible gamete, 1, 4, 2, 3, and 2, 4. And again, we could have the same combinations from this. And as we put these together, again, we'll do all this kind of stuff, and you just keep on rolling right through here. And again, you could fill out the entire Punnett square, and from this, now I can find the probability of what offspring are going to have which genotypes and which offspring are going to have which phenotypes. And again, the round variety we said is dominant, so if there's a heterozygous, it would still be round. So all of these would still be round, and all of these would still be yellow because they have at least 1 capital Y. And again, this whole Punnett square business is a way to look at a little bit of probability of offspring. Now, is everything so exactly cut and dry? And it's not, because we have codominance, we have incomplete dominance, we have some of these things being expressed, but the same basic idea. Out here is going to be the gametes. These are going to be all the possible offspring. When you start looking at this again, if it's homozygous, it means the same. We could take a look at this allele. If it's homozygous, it has both. It ends up looking like this. The phenotype is the physical appearance. The genotype is the combination of genes and the letters that go with it here. So heterozygous spotted and heterozygous outlined. And that's what it looked like. So instead of it having one or the other, it has both. Now this one would be homozygous outlined. And it has the outline pattern and no spots. Homozygous for the black allele, look like that. Heterozygous black and spotted, and it turns out all black. So in this case, you can't see the spots because the black is dominant over the entire thing, and even though it is carrying the allele for having spots, you don't see spots. It just shows completely like this. So heterozygous black and outline, again, in this situation, it turns out completely black even though it's heterozygous. So again, in this situation, if we have this outlined yellow and spotted yellow, the genotype would look like that. Same thing over here. And again, same thing here. And then we should be able to pick out what they're going to look like. And that's that. So in this case, we've got heterozygous black and outlined yellow. So it's going to be like that, but ultimately it's going to be like that. Because even though it's heterozygous, the black is still what actually is seen in the phenotype. But again, this whole idea, when it comes down to real phenotypes and real genotypes, you can have things that are not so cut and dry and simply this or that, and you can get all sorts of variety.